Hey guys, welcome to our channel. So we've been decoding the Bible change extractions, and this one is putting our method to the test. This one's putting my patience to the test. So basically, if we can get through this one, we can get through anything at this point. So it's tricky. In order. We've got lion changed to wolf, lay down changed to dwell, which I had an S there, but that didn't really affect, as I said in my past video, it didn't really affect our results. So many S's, it doesn't really matter if there's an extra S in there because we're not flip-flopping in every possible order. Now, if we were flip-flopping them in every possible order, that S would make a huge difference. Okay, but we're not doing that. So an extra S among all those S's didn't do anything to our results at all. And now it's gone. So you have lion to wolf, lay down to dwell. You have trespasses to debts. You have those who trespass against us to our debtors, mat to couch, and wine skins to bottles. All right. So by crossing off your L and your O off of lion and wolf, you cross off your L and your O off lion and wolf, and you're left with INWF. Yeah. Yeah, this is so we brought INWF down. Now by crossing off your LDW and your LDW, because they're alike, off of lay down and dwell, you're left with AIONEL. So we brought AYONEL down right here. All right. Trespasses to debts. If you cross off your TES and TES and debts, oh man, yeah, yeah, I need that board that Megan was talking about. But you cross off your TES and your TES, and you are left with RPASS. ESDB, and we brought our PASSESDB down. All right, our DB here. So then those who trespass against us changed to our debtors. So we have to cross off in order from those TOSE, and we crossed off O and our, or the T. We crossed off the T in those and the T in debtors. We crossed off the O in those and the O in our. We crossed off the S in those and the S in debtors. We crossed off the E in those and the E in debtors. We crossed off the O in who and the O in debtors. Now we crossed off the O in order from our first. We crossed off the R in trespasses, and we crossed off the R in our in order. Now our is completely eliminated. Then we crossed off the U in us and the U in our. It wasn't, but it is now. And you were left with HWOESPASS against the whole word, AGAINST, and S from us. So, and then D, B, R. And that's what we brought down. Man, we brought down, starting right here, HWHTESPASS against the whole word, and then S from the us, and then DBR that was left from debtors. All right, then Matt changed straight over to couch, so there was nothing to cross off because nothing was alike. And we have, this kind of got a little bit screwed, but you have Matt, and then we carried it down here to couch. And wineskins to bottles, guys. You cross off the E from wineskins and the E in bottles and the S in wineskins, the first one, in order, and the S in bottles, and you're left with WINKINSBOTTL. And that is what we brought here. WINKINSBOTTL. 5 minutes to get through that. Well, I said this is a challenging one. Now we're going forwards in genetics. All right, if you start with I, you get iGenetics, which is iGenetics, a molecular approach, 3rd edition. All right, that's a book, but it probably got hit. So then if you move on to NWF, you get National Wildlife Federation Genetic Engineering. So you got some genetic engineering going on, and I circled that wherever we're at. Right here, we circled NWF, National Wildlife Federation Genetic Engineering, because that's an interesting one to look at as we move forward, looking for the pattern within the pattern, and that's going to be another thing, guys. I don't know how this is going to work out for us, but moving on, you have AYO, and AYO ends up being AYOA Toye and Roger Cox, behavioral genetics. All right, behavioral genetics. Recent genetic mapping experiments in the mouse have made significant inroads into understanding the complex genetics of behavior and, in particular, anxiety. So our understanding of genes that influence behavior, including anxiety, is central to our quest to understand the genomic basis of variation in human behavioral pathologies. All right, and this goes on. But I'm going to move on, or we'll be here all day. I'll provide these links, guys. You can read all about this stuff. So then you have from AYO, then you have NEL, which ends up being the NEL gene. And it is a protein coding gene. And it's in chickens. Now, my fiance predicted going into this that we were going to find animal stuff in our genetics decode, and here we are. Chicken gene, protein. So, and that's NEL. Then RPA ends up being replication protein A in humans. All right, human replication protein A, RPA, is a heterotrimeric protein complex, was originally defined as a eukaryotic single-stranded DNA binding SSB protein essential for the vitro replication of simian virus 40 SV40 DNA. So it's an indispensable player in almost all DNA metabolic pathways. So, such as, but not limited to, DNA replication, DNA repair, recombination cell cycle, and DNA damage checkpoints. All right. So that was your RPA. And if you move on from RPA, you get SSE, which ends up being SSE1 and 2 gene and it's a yeast multigene family. New members of the yeast H7P70 multigene family. So we've had that. We've had yeast genes before, guys, in other searches. So that's not actually uncommon or surprising. Then if you move on from that, you got SDB, which ends up being Society for Developmental Biology. Now I have them circled because they come up again later. All right, and so we'll see how that goes. Then you got sleep disordered breathing. It's a genetic disorder. That's there as well. So they're both there. I might just provide that search. Or, you know what, I'll probably end up providing both individual links because that's really all you got. But moving on from that, you have HW, which ends up being Hardy-Weinberg Law of Genetic Equilibrium. And I'm supposed to read about that, I said. So the Hardy-Weinberg equilibrium is a principle stating that the genetic variation in a population will remain constant from one generation to the next, absent of disturbing factors. For instance, mutations disrupt the equilibrium of allele frequencies by introducing new alleles into a population. So that is a law on genetic equilibrium, and I will provide that. So that's HW. Then HT then ends up being stands for one of their things, one of their representations for genetic drift, which we've had before. We've got hereditary hemorrhagic gensitasia, and that is a disorder that results from the development of multiple abnormalities in the blood vessels. So you got a couple of things here with HT. All right. Moving on from that, you have ES, which ends up being ES cells, which ends up being embryonic stem cells. ES cells. We've had those before. All right. Then you move on to PA, and you got propionic acidemia, and that is a genetic disorder. Embryonic stem cells are cells that are derived from the undifferentiated inner mass cells of human embryo, ES cells. That's where we got that, guys. I'll provide that. But I wanted to go to, oh man, where was I? HT, and then ES, and then, PA, propionic acidemia, right here. Propionic acidemia is an inherited disorder in which the body is unable to process certain parts of proteins and lipids, fats, properly. It is classified as an acid disorder, which is a condition that leads to an abnormal buildup of particular acids known as organic acids. All right. So I'll provide that link, guys. We're going to try to get through this. Then you have SSA, which ends up being single strand annealing. Single strand annealing is a process that is initiated when a double strand break is made between two repeated sequences oriented in the same direction. Single-stranded regions are created adjacent to the break and extend to the repeated sequences that the complementary strands can anneal to each other. This annealed intermediate can be processed by digesting away the single-stranded tails and filling in the gaps. And this is all dealing with DNA and genetics, guys. Evidence for SSA in yeast was observed when it was found that HO endonuclease could stimulate deletions events between URA3 sequences in VIVO. All right, and this goes on, guys. You can read about it, but it's there. That would be SSA. Then you have You move on from that. You have GAI, which ends up being a GAI gene. I'm not even going to try to pronounce this word. I don't think Arabidopsis. Arabidopsis. GAI gene defines a signaling pathway that negatively regulates gibberellin responses. All right, and so this is on NCBI. We seem to come here a lot. So that's a gene. Then if you move on from that, you have NST, which ends up being NST1 gene, and it is a protein coding gene. We've got those before a lot. Actually, this is from the University of Minnesota website. And it's a worm-based gene. So there you go again, and more animal stuff that my fiance actually predicted would come up. And then you move on and you have SDB again, and that's Society for Developmental Biology. So that's twice they've come up in this search alone. So now they're circled here, and now they're circled down here. So we're going to look at them as we go as well. Then if you move on from that, and I broke those up so we didn't just get that twice, by the way. You have SD Genetics, and you have SD Genetics, which is a company out of Nebraska. If I remember correctly, they do swine genetics. But all I can find is their Facebook stuff now for some reason, but I think we've had them before. You also have Sanford Medical Genetics Laboratory in Sioux Falls, South Dakota. I usually stay away from the states' symbols or abbreviations, but in this case, a genetics lab in Sioux Falls, South Dakota seems important. You also have SD mutation. All right, so you got a few things that came up with that. And then if you move on from that, you have BRM, which ends up being BRM gene, and it is a protein coding gene. In the fruit fly. So there you go, another animal, an insect base, fly base. BRM is a protein coding gene in the fruit fly. We've had this before. Then if you go from that, you have ATC, and that ends up being ATC. That ends up being a genetic mapping. Physical and genetic mapping at the ATA/ATC locus on chromosome. So, genetic heterogeneity in ataxia. I'm not even going to try to pronounce it. AT. So, and it's to 4 types different genes responsible for this disease. So there's obviously that's a disease. So ATC ends up being a genetic mapping thing. All right, and I'll provide the link, guys. It's a lot of information at once, and I'm just learning all this stuff, so bear with me. Then you move on and you have OUC, which ends up being Ocean University of China Laboratory of Marine Genetics and Breeding. Oh, some more animal genetics that my fiance predicted. And Key Laboratory of Marine Genetics and Breeding at the Ocean University of China, OUC. So, yeah, I think that pretty much speaks for itself. You got a genetics laboratory, and I suppose we should circle them as well. because, well, actually, we should circle this genetics lab here, and we should circle OUC because they have a genetics lab as well. So we're going to keep an eye on those. And then if you move on from that, you got HWI, which ends up being Tay, HWIL Schwartz, PhD. And he's into studying, oh, what's that? What in the world did I do? There, I skipped one. My bad. Dr. Schwante's research focuses on understanding the genetic underpinnings of complex diseases such as substance abuse or substance use disorders and neurodevelopment/behavioral disease. And that's interesting because we got a behavior, we got AYO behavioral genetics, right? So now we got this guy down here studying behavioral diseases, and I will circle him too. I mean, wherever he's at, I've already circled him. All right. So now we've circled this guy, Tay HWI, Assistant Research Professor of Medical and Molecular Genetics at Indiana University School of Medicine. All right. So that's a pretty interesting hit. Two behavioral genetic hits in this alone. So then if you move on from that, you got NKI, which is Division of Molecular Genetics. NKI Division of Molecular Genetics, NKI, which is Netherlands Cancer Institute. NKI. So you have a cancer institute here. Division of Molecular Genetics. It's Netherlands Cancer Institute. So we're going to circle these guys as well. That's a lot of circled people, guys. That's a lot of... I can't really see what I'm doing. Let's just... then you have, if you move on from NKI, you have NSB. Yes, NSB. And NSB ends up being NSB Genetics Program. All right. And let's read a little bit about this. You have to come down a little bit. The NSB Genetics Program was developed in 2002 as a result of the blockage of the IWC quota renewal of the bowhead whale. So there you have it again, NSB Genetics Program, and these guys, and you have to come down. I'm going to provide this link, and you have to read down to genetic study to determine stock structure, and that's where it says the NSB Genetics Program was developed in 2002, and they deal with whales, so more animal genetics, just like my fiance predicted. Again, so then if you move on from that, you have OT, and OT ends up being SOX2-OT gene, and it is a non-protein coding RNA. We've had those before. Protein coding genes, non-protein coding RNA. And but this is a non-protein coding RNA, SOX2-OT gene. And I'll provide the link. Then you're down to TL, guys. That's your last two, and that ends up being MT-TL1 gene. The MT-TL1 gene provides instructions for making a molecule called a transfer RNA, tRNA, which is a chemical cousin of DNA. Transfer RNAs help assemble protein building blocks, amino acid, into functioning proteins. MT-TL1 gene provides instructions for making a specific form of tRNA that is designated as tRNA. During protein assembly, this molecule attaches the amino acid leucine and inserts it into the appropriate locations in the growing protein. All right. Did we get all that? And that gets you through going forwards in genetics. And we're at 25 minutes. So it's going to be a thing, guys. Maybe we'll have to do something a little different on the extractions. Maybe I'll just have to read them to you and skip the process here. Otherwise, we're going to go like this forever. But that gets you through forward. We got a few more things circled. Probably the most we've ever had to look at as we go. But I kind of expected that. This is the biggest one, most challenging one we've done yet. So the next thing to do is come back. And I'll get to that when I can, maybe tomorrow. Maybe, I don't know about later today. This one took a long time, and now we're at 20, almost 26 minutes. So I don't know how to cut those times down, but that's what you got. I will provide the links, guys, and um, that gets you through genetics. The next thing we will hit is medical. Or no, that does not. We will have to come backwards in genetics, guys. I'm used to this being the whole thing. We'll come backwards in genetics, and then we will go hit chemicals. So, yes. And that's going to be it. So I think we covered everything. So thanks for all your thumbs up, thumbs down. Thanks for your comments, leads, feedback, and subscriptions. And thanks a lot for watching, guys. And for now, you guys have a great rest of your day.